Hi, my name is Bryce Collingwood. Today I'm here to talk to you about my gut microbiome project for my Bio 200 lab. To start off, some background information. A microbiome is essentially just the microorganisms that are in a microenvironment. Typically it can be on your body, and generally they're just kind of around on the earth. Microbiomes are very important to humans for different reasons and correlated to our physical health. For psychological conditions, according to a book known as Brain Behavior and Immunity, microbiomes can help with alertness and consciousness. And basically this just helps with brain function, making sure that you're alert and conscious all the time. So having a healthy microbiome in your brain can help just being alert and help you keep being alert and your brain function at top speed. So our question and hypothesis for this experiment basically was we wanted to test the difference between two animals that were roughly around the same age but were completely different species. And then overall we stated that we believe that we were able to say the different species will have a majority of different bacteria in their gut microbiomes with some similarities, although there were some similarities between the two samples, the majority of which were different, and we'll get into that in just a sec. So yes, we were aiming to test the different ages of two animals, and I'll give you a short description of those animals. So we tested a crested gecko, and then we tested a common farm goat. Both were roughly around 6 months old, and then obviously the weight and height were very different, and the diets were very different. So the insect diet, or the gecko diet, consisted mainly of insects. It consisted of crickets and other sorts of bugs, and the goat ate mainly grass, hay, and sometimes ate vegetables. And then we'll get into our materials and methods. So starting off on our materials and methods, we started out by our three main steps were isolation, concentration, and sequencing. When sequencing is basically just telling what the DNA samples are. So starting out, we had the isolation of our DNA, of our fecal matter, and we took the fecal matter of both samples from the goat and the gecko, and we used a Kaigen kit. And we followed all the steps of this Kaigen kit. That is a commercial kit used to isolate DNA from feces. And basically what we did was we washed the feces through a series of wash buffers in a centrifuge to basically wash free the impurities of the DNA, or keep the DNA out of the stool samples that we could later use. Next, we move on to the concentration of the DNA after purification. We used for this the Zymo kit, which is another commercial kit used. The Zymo kit, we followed all the instructions again, mainly basically to the dot, just to make sure that we were keeping the concentrations correct. We made no changes to the protocol, but this Zymo kit, basically what it did was we took the DNA that we isolated from the stool samples and ran it through a series of different wash buffers to try and build up the amount of DNA samples that we had in our tubes that we would later use. So we then used our Amplicon PCR, and we were doing this to amplify the 16S ribosomal gene through the V3 and V4 regions of the DNA. We were doing this, we did this for 8 cycles, and then we basically used this at different temperatures to try and increase the amount of DNA that we were able to, using to quantify later in our experiment, which then led us to our gel electrophoresis tablet, which is right here. The numbers are a little small, but basically I'll explain. So our different samples, we had a positive and negative control here, along with our samples here and here. And then we used, excuse me, and then along with our other test samples, but we'll get to those in a second. Basically, what the spectrophotometer, or what the gel electrophoresis stated was that we were looking for if DNA was actually present in our samples, and these two lines here in our samples indicated that DNA was present in both of our samples. We were looking for a 550 base pair reading, which shows that DNA is present, and as you can see, 500 is here, and our readings are just above that, which signals that it's 550. And it ranged between 100 to 5,000 base pairs, and we knew that based on our reading, it was about 550 base pair readings. So we knew that DNA was present in the actual samples that we had. And again, just along with that gel electrophoresis, it was set at 250 volts, and it basically just separated the particles out by size. And 550 base pair size reading is basically how you tell what DNA is, because that's the size, generally what it is. Moving on to our next step, we took our samples and we ran them through what's called a PCR cleanup. In this, we used a commercial kit known as the Ampure kit for PCR. And what this did was it washed the DNA samples through a series of wash buffers, and we followed all the instructions here on this. And this removed the magnesium, the DNTPase, and the enzymes that would interfere with the indexing PCR samples afterwards, because it binds to the primers, which then have a hard time being quantified later on in the data analysis. We ran through the PCR cleanup twice, once initially before we decided to do the indexing, and then we indexed, which I'll get to in just a second, and then after indexing we ran it through again just to clean up any impurities. So like I said, we went through the indexing PCR after the PCR cleanup, and basically what we did here, we did 8 cycles again at a different number of degrees. So at the hottest degree, we this is when our DNA was denatured, and then basically we were able to kind of like slide different codons and primers into, not primers, codons and tags into our amplicons for each sample, and these acted like a label so that we could then once later on in our DNA analysis, we could tell which gene was amplified and which gene was present based on this the amplicon tags that we added to our sample. So basically that's just what the indexing PCR would do. Like I said earlier, after that we performed another round of PCR cleanup to take away any impurities that were left in our samples. This was just to make sure that we were dealing with pure DNA, and we didn't want anything else to kind of interfere with our sequencing, which would happen in our next step. So after our second PCR cleanup, we quantified the DNA of our indexing PCR products, and this basically just used a fluorometric assay, and this basically is another spectrophotometer instrument, but it uses fluorescence light to detect the tags that we added in indexing PCR, and then these tags were like the fluorescence from the machine is actually much more accurate at picking up the tags that we established in the indexing PCR, and it's much more effective than using a spectrophotometer, which can only tell concentration based on absorbance readings through light. But the fluorometric readings can actually tell which tags are present and which ones are not, and in which abundance. So it's much more effective. Our final step, once we had our quantification of data after the fluorometric assay, we used the Illumina MISSEQ next generation DNA analyzer. And basically what this does is it takes the DNA strands that we have isolated, quantified, cleaned, and then indexed and tagged. And what it does is it puts them in the machine, and basically what it does is it makes millions upon millions of copies of these DNA strands, so that we can get a good kind of like general sense as to like what DNA strands were in there. Obviously, the more base pairs, the more room we have to kind of like do experiments on, figure out which genes are present in which base pairs. So like I said, the more base pairs, the better off it is for our experiment. So we can move on to our results then. Our initial spectrophotometer readings for our 2 samples are here, our 260 to 280 ratios, which basically determine whether or not we were dealing with pure DNA or pure RNA, pure DNA or proteins. And all of our absorbances were almost right at where pure DNA is. 1.8 is what the pure DNA reading is for here. These were our concentration readings calculated from our spectrophotometer readings. So obviously some samples were a lot more highly concentrated than others. And then we actually had to calculate down our values so that we could use it later on in our reaction. We needed values that we could calculate down into different units so that we could make calculations later on. For concentrating our DNA, we actually had an error take place in one of our samples as we were using a spectrophotometer to calculate. The reading would actually not, the machine wouldn't read our values that we had, and so we couldn't actually take the value of our concentration through our nanomolar samples. However, the rest of the concentrations from our spectrophotometer are written here. We had 2 values that we couldn't be calculated because it wouldn't read our nanomolar calculation values, and we actually did not even need to measure our negative control value. Moving on to our gel electrophoresis results. This is the same table that I showed you from before. Our readings showed that our two samples did have DNA present along with the 550 base pair readings. Again, that's just showing that DNA was in fact actually present in our samples, which is good because that's what we needed to analyze in the first place. The quantification of our cleaned up amplicon PCR results are shown. Oh, I'm sorry. Actually, I showed you that already. Moving on. After we did our quantification of our results and our, so we have our concentrations, we have our purity readings, and we have all the information we need. We sent them the DNA through the sequencer, and the sequencer measures the order, the kingdom, and the genus of our samples. That's what we were looking at in general, the different bacteria within. This is our kingdom graph reading. So the blue columns here are bacteria, and the orange columns here are unclassified. So the kingdom of our readings for our DNA were for the DNA of our samples, the kingdom that they were part of. The DNA or the bacteria was upwards of 98%, and we had a very, very small area of unclassified that was obviously exactly that. It was unclassified, so therefore we could not basically tell what it was. Moving on to the diversity in our genus samples. The genus samples here are, again, different forms of bacteria that were found in both samples. So here we have the genus, we have Clostridium, Blautia, Escherichia, excuse me on some of these pronunciations, Sarcina, and this was our unclassified column that the machine could not actually read here. And then the rest of our bacteria here, as you can see through some of the samples, blue is 716, which was our goat sample, and 587 is the orange here, and that was our gecko sample. So there's a different overlay in some of the bacteria that is in their gut microbiomes through the genus order that we were looking at. And then moving on to our third graph, which was the order of our actual of the bacteria in sequence samples. And again, 716 is blue, orange is our 587, and it just shows again that there's a little bit of differentiation in the bacteria in the order of the microbiomes of both animals. Along with these graphs, we have down here a table here giving our samples and the number of DNA reads that the sequencer was able to calculate. Upwards of 2,000 reads is what we call successful, and that we can use to actually say that we believe our results are accurate to the point. So one of our measuring samples that we were using that was not ours was 555, and it didn't have the appropriate number of reads, and therefore we could not basically justify that the DNA was the DNA that we were looking at was the right DNA. However, the DNA sequencer did get 100% quality of reads, and this basically just means that of the reads that it took out of here, so the 83,031, the 83,000, the 3,000, and all below, the quality of the reads that it took were 100%, meaning that the DNA that they found, so the DNA strands that it found were in fact the correct DNA strands for the different samples, and that they were 100% able to quantify and basically track to make the DNA the same. We did have categories, like I said in our graphs up here, of unclassified, and we did have an other category. The unclassified sections of the reads were composed of bacteria that the sequencer could not decide on what category the species of bacteria fit in. And basically this is just, if the sequencer found a species of bacteria that it was unfamiliar with or wasn't sure necessarily like what to classify it as, it would put it in an unclassified category. It would put it in just kind of with the rest of the different types of bacteria, because it itself doesn't know what to classify it as. The other categories that the sequencer read was if there were bacteria or there were viruses or any other specimen that cannot be categorized as bacteria. Obviously, as thorough as the washing and purification of our DNA samples was, there's always room for a slight bit of error. If any bacteria had actually, or if any non-DNA samples that could have contaminated our specimens actually got in, this could have been an easy cause as to why there was an other category in our readings, and then this would be again why the DNA readings have an other category in their graphs. Moving on to our discussion of our DNA extraction from our scat. In terms of the quantity of DNA that we actually got out, it was successful. We did collect a large amount of DNA from our samples, and there was definitely a high enough concentration after going through our different concentration steps through the through the Zymo kit, and then when we were doing the PCR cleanup reactions, we were cleaning out large amounts of DNA, which is great, meaning that we had a high level of concentration that we could use later on in our DNA quantification. The spectrophotometry results showed again just that we did have enough number of reads, and that our purification of those the purity of those reads that we were using was very adequate for our experiment, and our concentrations that we used definitely were adequate to test and keep going through the rest of our experiment. So our Amplicon PCR results, as seen in the gel electrophoresis over here, definitely was very vital to our experiment. It showed that we do actually have DNA present in our samples, and it shows that like I just said, it shows whether or not DNA is actually present in our example. In our positive and negative controls, in our positive control, we did have DNA present. However, in the negative, it was not, and the negative control did not actually have any DNA put into it. For this exact reason, we wanted to have kind of like a test band to see what it would look like, and if whether or not the DNA would show up as present or not. And again, like I said before, at 550 base pairs, the level of DNA through the orange bands shows that it was in fact actually present, which was good because it means that we had DNA to quantify later on in our sequencer that we could use to show the different number of bacteria that we were looking at, and then that would be able to tell us which bacteria or which microorganisms were actually in the microbiomes of these animals. From our indexing PCR cleanup, down here at our concentrations were at a very high value, or a very good value that we could actually read. Like I said, the samples here, the concentrations were adequate enough because they were above 4 nanomolar, and that's what we wanted to get. The 4 nanomolar is kind of like where we wanted to be, and we were able to dilute these concentrations to the desired concentration that we needed. So we could dilute them down to 4 nanomolars that we had room to kind of like wiggle with. If we were to kind of screw anything up, we could always have extra DNA to work with. Our negative control was expected to have no DNA, which we thought would be true until we got back to the sequencing data. The negative control had 21 reads with multiple bacteria present, and this leads us to believe that there was cross-contamination between our experiment at some point. Moving on to our sequencing data, down here, our positive and negative reads was, the positive number of reads for our positive control was 56,622 with 100% reads passing quality filtering. There was 99.98% bacteria. Like I said, there was very, very small amount of unclassified or other. A lot of it was bacteria, which is good because that is what we're trying to discover in the microbiomes. At the species level, there were 97 total classifications, the main being, excuse my pronunciation, but Escherichia, which is only 0.03% of our total reads. And this tells us that there was most likely some type of cross-contamination somewhere along the process, or if other groups got generally the same readings, there was something wrong with the original sample. Basically, this tells us that for both our samples of interest, we got enough successful read to have adequate analysis, which basically means that the total, like I said earlier, the total number of reads meant that we could take those numbers and analyze the amount of DNA without having to worry about there not being enough reads to basically back up our results that we are kind of like using to prove or to kind of like support our hypothesis. And our last discussion at the time was their kingdom, order, and genus graphs. Both samples at the kingdom level were the majority of bacteria, like I said, up in the 90% range, meaning that there was very, very small, the majority was a very small percentage of the rest of the different organisms. The main difference in order classification is the amounts of bacteria we see in the sample of 716. The majority of that bacteria was at 71%, followed by the other form of bacteria at 15.98%. In 587, the one form of bacteria was also a higher majority at 37%, but the other bacteria was also present in 33.17%. So it just shows that there was a higher value in different species of bacteria. In the genus, we had there was a major a difference in the bacteria that we have in our samples. The only matching genus classification is the Blautia, which was around 16 to 18%. The other bacteria, the Clostridium, is the leading genus in 716, and the Bacteroideus in 587. The Clostridium are important in modulating the psychological, metabolic, and immune processes. It plays a large role in maintaining our gut homeostasis. And according to the articles that we researched along to kind of like understand why the gut microbiomes are important and what like our DNA would be able to help show, is that the bacteroids have been linked to inflammatory bowel disease. So an animal with high levels of bacteroids could possibly be linked to inflammatory bowel disease, and low levels of different bacteria forms, including the Bacteroides, can be linked to Crohn's disease, ulcerative colitis, and intermediate colitis. So basically, the lacking of different kinds of bacteria in the gut microbiome could be linked to certain health disorders, not just in humans, but in different animals as well. Obviously, the different DNA that would come from a human microbiome would be able to tell, like what kind of diseases could come from different forms of bacteria. However, for these two animals, like I just said, the species of diseases that could come from would be from the lack of bacteria that we have. Ladies and gentlemen, thank you for listening to my presentation on our gut microbiome project. I hope you learned a little bit about what we did to quantify DNA and figure out what microbiomes were in animals. Thank you very much.